Welcome to my channel MK Mediguide Lectures, learning with artworks like drawing and calligraphy.
In this video, we will discuss about the cytokine storm and immune senescence in aged COVID-19 patients. Immune senescence is described as the progressive loss of all immune effectors and functions in both the innate and cell-mediated immune system with aging. A normal and physiologic immunity depends on effective crosstalk between innate and adaptive immune systems. So, senescent immune effectors markedly impact on the health of elderly individuals. Here, we are going to briefly discuss age-related alteration of both innate and adaptive immune cells. Macrophages are central effector cells of the innate immune system and have many physiological functions. Macrophages can produce several pro-inflammatory molecules like TNF-alpha, interleukin-1 and interleukin-6, and anti-inflammatory molecules including interleukin-10, TGF-beta, acute phase proteins, and also bring into being enzymes, growth factors, nitric oxides, and toxic reactive radicals. As the clearance of pathogens proceeds, the anti-inflammatory factors of macrophages turn off macrophage inflammatory activities. During aging, the generation of several macrophage-induced factors is reduced, including TGF-beta, toxic free radicals, various growth factors, and expression of nitric oxide synthase. Additionally, phagocytic and chemotactic activity of macrophages also decreased with advanced age. It is reported that with progressive age, the expression of MHC type 2 also decreases in humans, which are required for effective crosstalk between innate and cell-mediated immune system. Natural killer cells are another cytotoxic factor of the innate immune system and involved in the early and faster than T cells defense against virus infection. The natural killer cells also function as linking the innate and cell-mediated immune system. Natural killer cells regulate immune function by the generation and release of various cytokines. An increase in numbers of circulating natural killer cells reported during aging. One of the important cytokines for cytotoxic activity of natural killer cells is interleukin-2, which increases killing properties and proliferation of natural killer cells. In a healthy, young individuals, interleukin-2 can induce interferon secretion by natural killer cells, but this effect decreases in elderly adults. Dendritic cells are another component of the innate immune system and have a critical role in immunity. The resident dendritic cells are immature, but the capture of pathogens converts them to mature form known as antigen presentation cells or APCs. Then mature APCs monitor T helper type 1 and T helper type 2 cells functions. Furthermore, dendritic cells can generate and release various cytokines, so they can modulate inflammatory responses. It is reported that increased age-related pro-inflammatory cytokines can induce activation and maturation of dendritic cells. Moreover, the activity of dendritic cells in elderly adults are higher than young individuals. Besides, aging is also accompanied by profound and consistent alterations of T cells' immunity. Although T cell numbers do not diminish during aging, however, T cells exhibit potent age-related alterations, including poor T cell mitogen responses, an inverted CD4 plus to CD8 plus T cell ratio, reduced proportion of naive cells, as well as an increased proportion of memory cells in older individuals. Additionally, aging is related to the overproduction of pro-inflammatory cytokines by T cells. It is demonstrated that aging also changes the profile of T helper type 2 cells, cytokines like interleukin 4 and interleukin 10, rather than cytokines of T helper type 1 cells, which include interleukin 2 and interferon gamma. These changes lead to a mild age-related inflammation in elderly adults. Aging can also potentially affect other T helper cells. The ratio of T helper 17 cells to T regulatory cells increase during aging, leading to a basal inflammatory stage in elderly adults. T helper 17 cells have the pro-inflammatory phenotype, and they are in balance with T regulatory anti-inflammatory cells. These cells are derived from a common precursor. Furthermore, during aging, naive T cells show multiple alterations, including the reduced production of interleukin 2 and the diminished ability to differentiate themselves into effector cells due to the reduction of proliferative capacity. Humoral immunity, mediated by B lymphocytes, has a critical role in the modulation of adaptive immune responses. B lymphocytes produce different types of antibodies for eliminating of challengers. Additionally, B lymphocytes have an important role in the immune system through the presentation of antigens and secretion of cytokines. B lymphocytes arising from hematopoietic stem cells in the bone marrow as pro-B lymphocytes. Then they differentiate into pre-B and then B lymphocytes. Ample studies identified that aging is accompanied by a quantitative and qualitative alteration of B lymphocyte pool. Production of precursor B lymphocytes in the bone marrow, and the number of pro-B and pre-B lymphocytes decreased in aged humans. It has been shown that aged pro-B lymphocytes have an impaired ability to respond to interleukin 7. Also, both human naive and IgM memory B lymphocytes impair during aging. Besides, the total numbers of B lymphocytes decreased. Additionally, the antibodies level is decreased in the aged subjects. The decreased function of B lymphocytes during aging has been attributed to lack of T helper cells function, because the function of B lymphocytes is T cells dependent. Taken together, immune senescence alterations cannot properly fit cell-mediating and humoral immune response in elderly adults. The immune system appears to maintain a mild inflammatory state in elderly adults. Therefore, it is suggested that fragile and mildly overactive immune system in elderly adults cannot turn off the pro-inflammatory mechanism in COVID-19 infection. Several manifestations, including lymphopenia, reduced numbers of CD4 plus T cells, CD8 plus T cells, B cells, and natural killer cells, monocytes, eosinophils, and basophils reported in severe patients with COVID-19 infection. And at last, thanks for watching our video and support us by subscribing our channel. And do like and comment below about this lecture.